All right, so this technique that we're analyzing here is called SDS. It's a specific type of gel electrophoresis that we're going to use on proteins. So the first step for doing an SDS gel electrophoresis is that we want to denature our proteins. It's not going to be very helpful if we have our entire protein sequence all jumbled up together, because we won't be able to look at each individual protein, which is kind of the whole point of running this thing. So, yeah, and then the next step is that we want to boil it in SDS. So SDS is an extremely negatively charged species, and so by putting all of the protein into the SDS, we're making it so that we kind of level out the playing field, so that we're not looking at all for charge. Like, we don't want to be sorting out the different proteins based on charge. We're only trying to sort them out by size. So by putting a very negative, uniform charge, we make sure that we're not actually like sorting anything out via charge, which is desirable for this. As you can see, this is kind of what it would look like. We have a little protein, and our negative charges are just engulfed around it. Okay, so that's the setup that we're going to be using for the SDS. And then after that, it's just kind of like a normal gel electrophoresis. So we've got our negatively charged part and our positively charged part. And we know that after we add the buffer and the sample, that the faster molecules, or the ones that moved farther, are going to be the smaller ones. And the bigger ones are going to stay over here, because there's more tension as it's moving across the gel, so the small ones are going to migrate a lot faster.